Hello, welcome everyone to the Precision Medicine Seminar. It is my pleasure to introduce our guest speaker today, Dr. Nita Limdi, who's visiting us from the University of Alabama at Birmingham. Dr. Limdi has actually 3 degrees. She has a doctorate in pharmacy, Ph.D. in epidemiology, and master's in public health. And she's affiliated with 4 different departments at UAB, truly reflecting the interdisciplinary nature of her work. So she's affiliated with neurology, I believe medicine, division of nephrology specifically. She's also affiliated with the Department of Epidemiology and Clinical Pharmacology and Toxicology at UAB. She serves as a director of the program for Translational Pharmacogenomics at UAB. She also co-directs the UAB CTSA, especially she's in charge of the training program, the training academy over there. She's also an associate director of the Hugh, Kaul Personalized Medicine Institute at UAB. And her work really spans across many different domains of precision medicine. She got started in pharmacogenomics. She's done a lot of work on the pharmacogenomics of coumadin and anticoagulation. But more recently, she's also working on polygenic risk scores, as well as on an implementation of genetic testing, especially in individuals underrepresented in biomedical research. She participates in a large number of precision medicine consortia, including Clinical Pharmacogenetics and Implementation Consortium, CPIC. She's also contributing to the pharmacogenomics knowledge base. She was involved in the International Warfarin Pharmacogenomics Consortium, and she's also part of other large NHGRI consortia such as Ignite and eMERGE4. And I know Dr. Limdi specifically from the eMERGE4 consortium work, where she has done actually an amazing job recruiting patients for polygenic risk score prediction and testing. Actually was #1 recruiter across the entire consortium of 10 different sites across the nation. And the first one to complete the recruitment. She's just really done an amazing job with this and has been a very close collaborator on polygenic risk modeling and CKD and type 2 diabetes. So welcome, and I hope to learn a lot from your talk today. Thank you.
Thank you for that very kind and generous introduction. Good afternoon, everyone. So I'm going to be talking about pharmacoepidemiology, pharmacogenomics, chronic diseases, and share, what we have been able to achieve for underrepresented and minority populations. So I started my career as a hospital pharmacist, and I wrote my first NIH grant with no mentors, and I submitted it to NIH, and they said, well, we don't know because you don't have academic pedigree. And I said, well, can I apply? And they said, yeah, you can apply. So this is the last 20 years, that was 20 years ago, of work, and there has been a tremendous amount achieved, and it really speaks to the team that has helped make this happen, and the mentorship of senior leadership at UAB. So my work focuses on chronic diseases. As a pharmacist, that's what I dealt with. They are common, they are complex, they are expensive. Our healthcare budget, 90% of our healthcare budget is actually spent on chronic diseases. And that for medications, that translates to about $410 billion a year. What you see in the graphic here is the dollars spent for common diseases at a per capita level. And what we get for that is a very, we don't necessarily get efficacy and the therapeutic response that we expect to get. Why is that? It's because we use a one-size-fits-all approach. I know institutions like Columbia and Penn and Harvard, these Ivy League academic institutions are doing genomics and are personalizing medication in multiple ways. But most of the patients you have hypertension, you are African-American, towards this calcium channel blockers and diuretics, right? So same diagnosis, gets the same prescription. And then while most people have therapeutic response, you do have a large proportion of patients that do have toxicity with or without benefit, and a significant number of patients who do not have therapeutic response. So for example, antidepressant, the first antidepressant you choose in a patient with depression has a 38% chance of remission, which means in close to 60%, 62% of patients, you don't see the benefit you want to see. That doesn't mean they have side effects, it's just they don't have therapeutic response. So this one-size-fits-all approach really does not work on this. Okay. So the program we have built really spans research, clinical care, and as Christoph said, I spent a lot of time. I have a K-24, so I spend almost a quarter of my time mentoring faculty in epidemiology, cardiology, nephrology, pharmacology. So really spend a lot of time helping them develop their careers. Our program goes from discovering predictors of drug response or disease risk to looking at their impact, to identifying their impact and effect size in cohorts of patients with the disease, to then testing them in larger patient populations and practice levels. And where we hope to go is at a population level. So we really are working along the T1 to T4 spectrum, and I'm very privileged to be able to share this work with you. From the get-go, so when I wrote my K award, I actually started writing it on anti-epileptic medications and Stevens-Johnson syndrome, because that was really not studied. And then 6 months into writing that grant, I switched and I said, but I really should be looking at warfarin. And my mentor said, well, there are other people doing it. And I said, they're not doing it in Africa, right? All the data was from patients of Europe. So, we have maintained fidelity to this original goal in everything we do. And we'll share some of this work. So I'm going to start with research. As a pharmacist, what was near and dear to my heart was adverse drug effects. They are the fourth leading cause of death. 2 million hospitalizations, $130 billion in healthcare costs. Very common, very expensive, very prudent. The biggest culprits, and if you look at data out of the CDC for the last 3 decades, the number one culprit that has not changed are oral anticoagulants. And this is true even with the newer oral anticoagulants. So it wasn't just warfarin. And so, this was a great place to start, right? And so we started our work in patients who were initiating oral anticoagulation and built prospective cohort studies. And this includes then atrial fibrillation and VTE cohorts. We enroll patients who are starting warfarin, starting the bigger trend, followed them for up to 2 years. And all their clinical events were captured, whether they happened within our system or not. This allowed us to begin to look at common and rare genetic variants that influence warfarin response. Really, we started with race, self-reported race, which we know is a social construct, but that allowed us to refine our, to identify genetic markers that were associated with African ancestry and how they affect drug response. It also allowed us to look at comorbid conditions and co-medications and the interaction between the two. And finally, it allowed us to look at diet and lifestyle. And, with warfarin, because it's a vitamin K antagonist, your dark green leafy vegetables matter. There is seasonal variation in intake of these vegetables, and the variation is higher in patients of African descent, at least in the South. So with this work, our goal is to really build How do you use all this information to guide dosing, to predict risk of hemorrhage, and to really personalize the risk-benefit trade-off, treat or not treat? So we built prediction models. We have looked at very deeply at kidney function and how it affects drug response. What you're seeing here is the biggest trend. It's not very widely used, but it was the first direct oral anticoagulant. And what we showed is it's not just patients with end-stage renal disease on dialysis. It is patients who have GFRs less than 60 that have a higher risk of bleeding. We have looked at race and everything that it is associated with it and how it affects risk of hemorrhage. And so if you go back to the warfarin trials, race is not mentioned. It is not in the main paper. It is not in the supplement. It is not in the FDA submission that the company did to get the drug approval. It is nowhere in the application. So patients of African descent have 60% higher risk of bleeding complications. This is after adjusting for clinical factors, for medications, etc. And then this finally has led to ancestry-specific guidelines in pharmacogenetics, where we have said that if patients have African descent, you make sure you are testing additional variants that are specific to African ancestry before you use it to make therapeutic decisions. And I will show you results of a randomized clinical trial to show you why. So being in the South, it was an opportunity to really look at genes and drug response and how drugs work or don't work in patients of different groups, and race is just one of the measures. So was invited to participate in the warfarin consortium, and it was really nerve-wracking as a pharmacist in a school of medicine with no safety net to share your data before you publish your first paper. But we did it. And so this, what you're seeing are the main results of this analysis of 5,000 patients. And what this graph shows is among patients who need 5 milligrams, that's kind of the dose you start people on. Adding clinical factors like height, weight, co-medications doesn't improve prediction of dose. Adding genes that influence warfarin metabolism and action doesn't matter. That is 54% of the population in the clinic. That means that almost half the patients, it matters that you add clinical factors, and it matters that you add genes. So based on this, we did a genome-wide association study, and we identified a new marker that sits outside the 2C cluster. By the way, this is a marker that also influences response to clopidogrel. This was the GWAS hit even in the antiplatelet work that Alan Schulden out of Maryland has done. So the allele frequency is about 25%. Almost half the patients in your clinic have 1 or 2 copies, and that is associated with up to 25% dose reduction. So common variant, big effect. In the meantime, so this was discovered in 2013. In the meantime, there was so much buzz around pharmacogenomics. There were articles in New Yorker and Washington, what is WSJ?
Street Journal.
Well, it's Wall Street Journal. Sorry, I didn't read it, and I'm like, oh, okay. So lots of press on the promise of pharmacogenetics. So NHLBI actually put together a consortium called the COAC Consortium, where they randomized patients before they got their first dose of warfarin. So these are patients who are newly starting warfarin. We enroll them, we genotype them, we randomize them to receive either clinically guided dosing, which is height, weight, amiodarone, etc., versus a pharmacogenomic algorithm, which had the clinical variables plus 2 genes. Overall, the aim of the study was to see if it improved % time in target INR range, because that's associated with bleeding. And then there were some pre-specified analysis by race and sex. So what you're seeing on the right is overall the study found that there was no improvement in percent time and target range using genetics. In patients of European descent, there was an improvement. In patients of African descent, we did harm. Okay. Why did this happen? How can adding more information and genes that we know influence orphan metabolism and action, why did that take away from the model? So what you see is what was in one of the supplementary tables. If you look at the algorithms that we use at initiation and revision of the dosing strategy, the pharmacogenomic algorithm performed well in patients of European descent, but the clinical algorithm performed better in. So this really created an onslaught of discussion in the medical literature, in the lay literature. And by then, we had built a large enough cohort to look at each of these markers individually. Until now, what had happened is because we had smaller cohorts and because these loss of function variants are less common, we collapse them as do you have loss of function or not, right? So lesson number one is the discovery of gene variants, so any predictor for that matter, really needs to be done in patients of African descent or in all patients, whether it's you're looking at kidney disease, whether you're looking at race, whatever you're looking at in terms of underrepresented populations. What this is showing that the most common loss of function variant, the algorithm reduced the dose in the COAC trial by 18%. But if you look at the actual data, and this was then supported by 2 additional papers, that yes, there is a 20% lower dose in patients of European descent, but there wasn't a lower dose required. So you incorrectly adjusted the dose when that wasn't needed. If you look at VCOR, which is the target protein for warfarin, statistically significant dose reduction is needed. However, there is a 10% differential. And then, of course, the new variant that was discovered in 2015 is not part of the trial, but you can see its effect. And I've already, so the other thing we have looked at is the control of INR in patients of different, in African Americans versus European Americans. And so this is not access to care. These are patients who are coming to the same clinic. They have insurance. They are treated by the same physicians. They are managed by the same nurse practitioners and pharmacists in the coagulation clinics. They spend less time in cardiac medications, variations in diet and exercise. There are lots of factors. So I was giving this talk, and a nephrologist said, well, have you looked at kidney function in warfarin? Like, warfarin is removed by the liver. The FDA says you don't need to adjust dosing. And he said, well, you may want to look at that. And the last 10 years have been spent looking at kidney function and anticoagulation response. So this is looking at warfarin dose in a adjusted model with an interaction term between genotype and GFR category. What you see is as the GFR decreases, warfarin dose decreases. This is a drug that is removed by the liver to inactive metabolites, renal clearances, it's not. And this is independent of the patient's of the genes that influence warfarin response. And the dose reductions at 10 and 20%. 40% of patients are affected. So we don't teach this in med school, that if you have a patient whose GFR is less than 60, consider a lower dose. Everybody has a fluid balance. Patients who have lower GFRs spend less time in target range. They have a significantly high risk of bleeding. So this is the dotted red line is GFR less than 45. and the solid red line is less than 30. But the package insert still says you can use warfarin without any adjustment. So as our cohorts increase in size, we could begin to add interaction models to survival analysis, pH-reg models. And so what this graph is showing is the GFR doesn't increase risk. A lower GFR is not associated with a higher risk of bleeding unless your INR is over 4. The issue is patients who have poor kidney function, they have more episodes where the INR is over 4, and when it is over 4, it declines slowly, which means they have a longer period at which they are at risk. So with this work, I worked with the FDA for 1.5 years. And one of the things I wanted to do, and they allowed me to do, is to get access to the clinical trial data for all the new oral anticoagulants. So they did not give me the ROCKET studies, which was rivaroxaban, but they gave me the other 3 AFib studies. And so this is data from the clinical trials with adjudicated endpoints. So I know clinical trials exclude patients who have end-stage renal disease or who are on dialysis. However, you can see across these the 3 AFib trials, 40%, 35, anywhere from 35 to 40% of people had GFRs less than 60. You can do something with this data. What you're seeing is these are the endpoints that were adjudicated by blinded adjudicators as part of the trial. So no surprise there. Warfarin, your risk of bleeding increases as your GFR decreases. Same for dabigatran, same for apixaban, and you can see it is less than it's GFR less than 45. The risk is higher. Same for edoxaban, right? So think about if you look at least at UAB, 20% of patients who had AFib had GFRs less than 45. For those 20%, their risk-benefit trade-off is not what it is for the patients whose GFR is greater than 60%. And so this really helped us as we planned our next study, which is the current R01 that we have, where we are building a cohort of 4,000 patients. Our aim is to predict risk of hemorrhage. We are looking at antiplatelet medications, vasoprotective therapies, do PPIs really protect you? Drug interactions, and then and mainly kidney function, and we are looking across the spectrum of kidney function. This is a rivaroxaban and apixaban cohort, and we embedded 180 patient PK/PD study. So I'm going to share some unpublished data, which we are still in the process of analyzing. But let's look at apixaban. We had a cohort of 1,700 patients so far. 40% are African-American. 1.2% of patients in the clinical trials had African ancestry. Okay. You can see the distribution of kidney function. 37% have GFRs less than 60, so pretty good sample size here. From that cohort, we actually built our PK/PD cohort. So we invited patients, 20 patients in each of these GFR categories for a 12-hour PK/PD study. And we didn't specify a dose. So, we ended up getting 20 patients who had a lower warfarin dose, lower apixaban dose. You can just see the distribution here. These are the findings. So on the left is 5 milligrams, and these are pharmacokinetic profiles. So this is apixaban levels. The solid line is GFR less than 30, not on hemodialysis. And the dotted red line is on hemodialysis. Okay. you can see a lot of the apixaban, dosing apixaban in hemodialysis because you're clearing the medication during dialysis, their concentrations are more in line with what you would see in patients who have relatively good kidney function. So what this is telling us in the PK analysis of pharmacodynamics, so 10A inhibition is showing similar effects. It's not finalized. But patients who have GFRs less than 30 and get 5 milligrams twice a day, they have really high concentrations. We see the same thing for 2.5 BID. These numbers are smaller, and note that, I had to change the scale to show the separation of the graph. So then we looked at major hemorrhage, and the FDA recommends if your weight is less than 60, if your serum creatinine is greater than 1.5, if your age is greater than 80, you should use a lower dose. Okay, so 9.6% of patients have weight less than 60. And yes, weight does matter because they have a higher risk of major bleeding. Serum creatinine is the biggest predictor. So GFR is the biggest predictor of risk, and age really did not matter. You can see if you look at less than greater than 80, or if you look at the 65 age cutoff, which a lot of leading prediction models use, you begin to see that it doesn't matter. And maybe it's because it's collinear with GFR. There can be many reasons for this. And so this is data we are hoping to publish that the biggest predictor is kidney function. It is not age. It is not, we use HAS-BLED, which is hypertension, age greater than 80, stroke, prior bleeding, labile INRs, end-stage renal disease. So most of these bleeding risk scores classify kidney disease as end-stage renal disease. But, perhaps we need a little more granularity and give points for GFR less than 60, less than 45, less than 30. And that's what we are hoping to do as we develop these models. So completely non-genomics pharmacoepi project. And we are going to add a genomic component. I have Jim, the faculty that's working on pharmacogenetics as part of that apixaban cohort. A lot of my time is also spent in thinking about how we translate this into clinical care. And we are thinking about this at a practice level. So if you think we started with cardiology, most of these patients get aspirin and clopidogrel after stent. 30% of patients have loss of functional leads, which means they do not have enough active metabolite to inhibit platelet aggregation. And this is evidenced by several meta-analyses that were done from clinical trials that showed loss of functional lead patients have a 60% higher risk of major adverse cardiovascular events and are almost threefold higher risk of stent thrombosis. And there are alternatives that do not go through that pathway. So our first implementation of pharmacogenetics at a practice level was really beginning to do CYP2C19 point of care testing. There is a Canadian company, buccal swap to genotype in 60 minutes. And it only tests 3 variants, but evidence is on those 3 variants. So we started testing, and for patients who had 2 loss of functional leads, we did require them to ask the physician to change to ticagrelor or prasugrel. And then we followed the patients for a year. And, by this time we had had a track record of working with consortia. So these data were pooled across 9 sites that you can see on this graph. And what we showed is 31% of patients had loss of functional leads. That if you left the loss of functional patients on clopidogrel, 15 of them would have one of these events at a year. If you switch them to an alternative, 8 of them would have, 7 of them would have an event. So that is a 50% reduction in hard outcomes that CMS cares for. And we followed that up with a cost effectiveness analysis. And, you know, it is this kind of evidence that allowed CMS to reimburse for the test. And at least for our region, it is reimbursed at $291 a test. So this was, then the health system decided to support pharmacogenetics, and we now have a team. We are launching a more comprehensive approach in which we are testing 97 variants and 26 genes. And we are going to include other things like height, weight, kidney function, smoking, co-medications, dosing factors to help physicians optimize medication therapy. And this is a slide I showed the administrative leadership. So I show them that, you know, this is not just important for cardiology, it is important for cancer, it is important for immune therapies, it is important for psychiatry, and for infectious diseases. And our goal is to get to the right medication at the right dose the first time. Okay, so couple of examples that helped us convince. We had a very young woman who came to us after trying 3 antidepressants with no benefit. She's now on a 4th one at maximum dose with no benefit. And she comes to us for electroconvulsive therapy as an ECT referral. The psychiatrist decided to test her, and it turns out that she's an ultra rapid metabolizer on both of the pathways that metabolize to duloxetine. And so she didn't need a different drug, she needed a different dose. And yes, this is twice the maximum recommended dose on the package insert, but that's what she needed. She's remission free. A lot of institutions are doing somatic testing in patients who have recurrent cancers. However, even at UAB, we are not systematically doing pharmacogenetics to treat, to target dosing and medication therapy for the first-line treatment. And so this is something we are rolling out with colorectal cancer. And we have the added benefit of a panel approach because it will help us tailor that pain medication and antidepressants. And CMS, by the way, now covers for preemptive pharmacogenetic testing if there is an indication. So it's broadly covered by CMS. The way we have set it up is if we want to bill for the test, you cannot just say, I'm doing this panel test. Somebody needs to say, I am doing 2D6 and 2C19 because I'm considering Paxil and stuff. Right? And the lab doesn't know that. The gene drug combinations that you have to specify. So we have a Pharm.D team involved to help build this process. We are going to offer both point of care and a panel-based approach. And the PDX data is going to flow 2 ways into the EHR. One is the pharmacist is going to input this in Power Forms, which is a Cerner plugin that allows us to import things into notes as we write them. And then we have a live decision support system ACTEX. So in the future, when somebody enters clopidogrel on me, they'll say, use ticagrelor, she has to loss of use. So that's kind of how we are envisioning, and this is rolling out. This has just started rolling out. So we are looking at how we use this at a regional level through the Alabama Genomic Health Initiative. So here we have enrolled, now we are doing preemptive pharmacogenetics. So this is before the patient has a clinical need, because it is the most cost-effective way of doing pharmacogenetics. It's just that you may not be able to show return on investment or value because the patient may not have the actionability yet. And so initially AGHI was just doing an array. Then if there were hits on the ACMG gene list, then they sang or confirmed and returned the results. And what we ended up doing is we added pharmacogenomics to that, and we bring that back into seminar as both the PDF and the PowerPoint bill, so we can then use it for consultation. What this graph shows you that 100% of patients have something actionable. They may not need it yet. And what you're seeing on the graphic here on the left is across the different common genes that are involved in drug metabolism, how many patients have intermediate or poor metabolizers. These are the people who you would do something with. And then we have been working with Ignite to really look at actionability in both adult and pediatric patients. And this was more an effort to show that these drugs are commonly used, that their use is not decreasing. And that this is going to be a significant opportunity going forward. So one of the key things my mentors did is they made sure that I focused. Because when you have so many ideas, you can scatter your energy and not achieve. So what you're seeing in this graphic is over time, the blue bars are number of pharmacogenetic publications from 1990 to 2020, and the inner line graphs are publications in patients of different ancestry, so with self-reported race groups. They are not to scale, but it shows that over time, the pharmacogenomic consortia have really tried and have been successful in increasing the diversity of participants. And so I wrote my K award at the first Pharmacogenetics Research Network meeting. And with the completion of the Human Genome Project in 2003, it really propelled the field forward, not just in terms of what we could do, but the technology available to do it, like point of care. There are companies that are working on using your iPhone as a PCR machine and doing simple genetic tests on your kitchen table. There's a company called Biomeme that is working on sickle cell. So you can use it out in the field in Africa. So lots of technology companies are in this space. Then when CPIC was started in 2009, got involved in that. So we review the evidence and develop guidelines that are freely available. And they also create, as you begin to implement, they also create the plugins that you may want to drive your decision support and the text. Implementation of genetics in practice is really taking the guidelines and developing tools and frameworks for implementation. The Personalized Medicine Coalition, which is a think tank and an advocacy group for all things precision medicine. Then working with the FDA in the PGX space. And then finally, the Sprite, which is a collaborative community that looks at, that actually brings together all stakeholders, patients, labs, CMS, FDA, researchers, health system leadership, to talk about pharmacogenetics. And the one thing that was missing was work in common diseases, and Emerge really filled that gap. So I'm going to shift gears. This allowed us to look at the fourth iteration. This is the fourth cycle of Emerge, and this time they are looking to see, can we use genomics to predict future risk and intervene to one seed can be adopted, but the eventual goal is, can you use this approach to delay, attenuate, maybe prevent disease? So those are the goals of Emerge. Can we leverage this information to predict risk? Can we identify patients at high risk? And can we implement or use, we're doing PSAs before the age of 50 or additional testing or treatment, start a statin or intensify a statin in a patient with high CHD risk? And would that prevent, delay, or attenuate that disease risk? So Emerge is exciting and challenging because it is T1 to T4 in 5 years. Christoph can can attest to that. In years 1 and 2, our task was to develop and or validate polygenic risk scores for at least 10 diseases for all ancestral groups. And we started with 26. These 10 made the cut because we could have polygenic risk scores that could be clinical grade level and can be implemented in clinic. Our next task, which I think we have met, is to recruit 25,000 patients across 10 sites. Our goal was 2,500, and collect clinical data, family history data, genomic data, both monogenic and polygenic, and social determinants of health, and put this together in a comprehensive report. Put that back in the EHR, and our eventual goal is to compare collectively what did the high-risk patients providers do? Did they order the test? Did they do an earlier screening? And we are going to compare for recommendations more frequently implemented in high risk versus not high risk, or risk reducing interventions. Did that lead to new diagnosis differentially? And did it change any patient outcomes? Now, obviously, in a 5-year study, we can't study hard outcomes, but maybe we can study blood sugar or BMI, something like that. So we've had the pleasure of working with the CKD expertise here at Columbia to work on chronic kidney disease with Atlas Khan and Christoph, where we looked at developed in polygenic risk score for all 4 ancestral groups. And then we worked with Mass General Brigham to look at diabetes. We were asked to recruit more patients, and we have recruited about 500 more patients. And I am very, very proud of our team because our data completion is incredible. We have DNA on 98%, family history on 98%, and social determinants of health in almost everybody. So I think this will create a very rich data set, not just to do genomic work, but to do other analysis. So on time, right? I have time? Okay. So the type 2 diabetes work was done with Mass General, and here we use a method developed by Tian Gi at Harvard, a Bayesian approach. And what we did is we looked at large GWAS studies that were done in European, African, and Asian populations to develop our model. And then we evaluated these models and validated them in Emerge, phase 1 to phase 3. They have about 100,000 patients. In the cohorts that were at UAB that are African enriched, that were not previously used in GWAS studies, and the Taiwan biobank. And what we found is that patients who have that polygenic risk score is in the top 2 percentile have a 3 to 4-fold higher risk of developing type 2 diabetes. And so that's the cutoff then, is what the lab used to classify patients at high risk. This is work done by Atlas and Christoph here at Columbia, where we developed a polygenic risk score in 70% of the UK biobank, used and validated these risk scores in the remaining 30% in Emerge, in BioMe, which is a Mount Sinai biobank, and in the UAB cohorts. And this was incredible work because we tested this in all 4 major ancestral groups. And you can see here, the top 2% of PRS is associated with a 3-fold risk of developing CKD. And importantly, work done by Christoph and Atlas looked at the influence of PRS alone, ApoL1 alone, which we know is associated with CKD risk, and the combined effect, and really showed that it is additive, not just in patients of African descent, but also in patients of Hispanic descent. And I think this is the first paper that showed this effect. in Hispanic patients. So our next task, this kind of work was done for all 10 emerged phenotypes, and we are now in the process of enrolling and returning the results and building off our pharmacogenomic work. We already have the infrastructure and the teams in place to return these results. And we have, about 70 to 75% will not have a high genomic risk, either monogenic, polygenic, or integrated, where we notify every provider in the EHR via an impact message, and then the results are mailed to the patients. This is done by the pharmacist team. All the polygenic risk scores, we notify the patient, we return the results and explain the results in person to the patient before making the results, again done by pharmacists. And then the integrated and monogenic risk. Integrated risk is for breast cancer, are returned by genetic counselors. And we do have these embedded in reports and documents in Cerner. So they have, and we are tracking provider action. So at UAB, we have returned about close to right under 2,000 as of yesterday. And what you're seeing here is what we expected, about 25% would have a high polygenic risk, and we are kind of there. You can see the monogenic risk is around 4%. We had estimated this would be 2.5%, but remember, we don't sit on monogenic results. So if we have, I think we have about 2,800 JIRAs that are kind of in the process of being generated, we prioritize. So that number should go down. And then another 2% have a integrated risk. And we are beginning to now make headway on the proposed results. So we are looking at what if the patient did? Did they stop non-steroidals if they had a high CKD polygenic risk score? We are also surveying the providers. So we have really focused on representation, and I will close by looking at a global snapshot. So if you look at 1950s, right after World War II, 22% of the global population was Western Europe. So think about where the genomic, what population is best enriched for genomic data. That population today is 10% of the global population. And in 25 years, it will be 7% of the global population. So we really need to do better for the 75%, which is patients of African descent and patients of Asian descent, to really understand their disease risk and their response to medications. This is the same kind of look at demographics in the US. Our demographics are changing, and we will actually have more patients of of Hispanic descent, and Asians and African Americans will also be increasing. So I think our task ahead is looking at the population changes and making sure that our research really addresses these underrepresented populations, whether we do it by race, demographics, kidney function, whatever it is. And I will end by thanking the incredible team that I get to work with. We have a core team that does the recruitment and retention across my studies, that does the data curation and quality control. The clinical pharmacogenetics teams, they work on different projects, but that actually helps them do better for each project because they share solutions. And I've worked with the statistics team for close to 20 years now. Bruce Core, people at the FDA that allowed access to that, the clinical trial data, and then the Emerge and Ignite Consortium, especially investigators here at Columbia, Harvard, and Sinai that have worked closely together. And behind all these people, I know there are even more people that have done it. So thank you.
Questions?
Yes, Don.
Great talk. So I guess my question is related to the race perspectives that
Yes.
let's say basically if we define race, right? Biologically, we should using genomic information to define race, right?
Yeah.
But when we conduct a study, are we saying, okay, this race is a reflecting of the more of the biological, let's say genomic part, or actually it's from the cultural lifestyle? Because sometimes they are highly correlated. Is there a way we can actually look into whether, it's the genomic contribution or whether it's the lifestyle, cultural contribution?
So race, the way people self-report it, is really a social construct. And as, I don't know what if you look at people who say I'm African-American in New York, what their African ancestry is. But I would say it probably is going to be lower just because of more admixture than it is in the South. So when we did our orphan work, and most of the genomic studies have really started with self-reported race. So it's a patient's identity rather than their ancestry. And yes, it has been correlated, but we cannot rely on that as more admixed. It will not be. So in our warfarin work, when we looked at it, it was somewhere between 86 and 87%, from the genetic ancestry perspective, was Western Africa. So it's a very complex construct. It is a very politically charged discussion point that will take, and the concept of race really has evolved over decades and has influenced and is influenced by the politics of our time. And it's going to take very focused, very uncomfortable discussions for us to really get to a point where we understand how to use that. And I know Emerge Prime Consortium is attempting to do that, as our main groups on what does that mean and how do we capture all the aspects of race. So, when in my warfarin work, what I noticed when I looked at African patients who said I'm African American or Black, their diets were different. Their vitamin K intake was different. Their vitamin K intake, in the winter when they ate more collard greens and turnip greens, their warfarin dose changed, right? And that actually had an effect, probably more than their genes did. Physical activity influences your basic metabolic rate, that influences drug metabolism. So there may be, it is a very complex construct. Race as is self-reported is really more to do with identity than ancestry.
Trying to understand what will be the future in terms of when we, let's say, get race involved in all the research designs. Are we saying, okay, we are still going to rely on this self-reported race, or we are trying to understand more from genomic or biological perspectives?
So I don't know if it is either or. I would say it is both, because your genetic ancestry doesn't capture everything. Doesn't capture culture, doesn't capture your social network, doesn't capture so many things. So I think it is both. And I don't know how we do it. I think that's what is being discussed a lot in lots of forums, not just in genomics, but even in pharmacoepi. And how do we look at this?
I have wonderful studies, and I'll speak a little louder so that people around the corner can hear me too. Great studies, and it's all wonderful. And then you come down to patient care, for instance, at an institution like here. And if you look at all the pharmacogenomics tests here, they're 0 to none. And it's not for the lack of trying for various people. And this is just here in New York City. But if I look around the world, you have certain places where the implementation goes a little bit better, but not as optimal as you would like it to see. Now, what would you give us advice on how to implement, better implement this? What are the major hurdles that we really need to clear?
So implementation is hard. Everybody talks about it takes so many years for a drug to make it from phase 1 to market. It takes evidence on average 17 years after you have published, it's been validated, replicated, people acknowledge it to implement. Because implementation is about behavior change. And we started these discussions about pharmacogenetic implementation in 2008. We are now implementing. COVID did throw us off by 3 years, but it is going to take time. We also had to, work. The consortia have been very powerful and impactful in this area because they have really done rigorous work to show the evidence and the effect on outcomes that payers care. So implementation will take time. I'll give you an example. In 2016, in the last, what year is it? 2024. 2016, 2017, the stethoscope turned 100 years. There was an editorial in JAMA. Anytime I get disillusioned, I read that. Do you know what it took to get the stethoscope to be used by physicians? It was unsurmountable. Because physicians said, why do I need this? I can put my ear and hear the heartbeat. Can you imagine medicine without the stethoscope? But, it is going to take time to implement. As we discussed at a meeting, is implementation needs support from top down. So you need the leadership support, but to really get it to work, you need the buy-in from the people in the trenches. And so if you just go top down, you will have a lot of resistance. And, we started, and that's where my experience and connections as a hospital pharmacist team became useful because it allowed me to say, okay, we started in the cath lab because it is an interventional cardiologist, right? So don't start with statin where anybody, all the patients go to one physical location, which means now you can train nurses to say, this is how you collect, this is where you see the sample. They know how to do this, right? But still, it's something new. And you need a champion who's going to shepherd the process. And that's what it is. It is very beautiful. It is so much easier to do research.
This was a great talk. So quick question. I think one of the opportunities for pharmacogenomics is integration of EHR and developing principal decision support based on genomic data that is still structured format in EHR. Yet our EHR systems are clearly not designed to handle this, or it's been really difficult, at least at Columbia, to implement genomic data storage within EHR. You mentioned that for some of your studies, the genetic variant information is actually stored in Cerner. Could you share some experiences about how you got this done and, perhaps outline how it works?
Yeah. So you guys have Epics?
We have Epic.
Okay. So Epic actually has a pretty good genomics module that people are using for storing discrete data. Cerner is, how shall I say, infinitely flexible, which means every problem needs a new solution, right? Because there is, So when we started the CYP2C19, we had to get the bill done by our health system informatics people to store that information as discrete data. With there are companies that do decision support within Cerner and Epic, like Actax. So what happens is what we are going to do is as we build that footprint internally, we will use Actax. So the data will go from UAB's pathology lab to Actax. And then when a physician, so there will be a PDF, there will be the power form, and then Actax is what will trigger the live decision support. So in the future, when a physician clicks on a medication for which the patient has a pharmacogenomic actionable variant, that will trigger whatever alert we want it to pop. So you can personalize these alerts. And so there are companies like Actax, there is Admera, there is, it was in Nita now, in Nita is being bought by Ford. And, but there are companies that do this live decision support that can help you. The disadvantage is it's a subscription. So you have to pay, so, you know, and there are, whatever the rate is per year for up to X number of tests or X number of alerts. But within, I don't know enough about Epic because we don't have Epic, but I hear that these data can be stored discreetly. And then you build the decision support internally according to CPIC guidelines. And CPIC actually has the plugins that you can copy and paste and build it.
I can
That's-
just tell you it's not as good as it sounds, so we
Okay.
have a genomics model that's not quite functional, but yes.
Okay.
There is a way forward. I just thought maybe you had also solutions in Cerner. But it sounds like you're using an external.
We are using an external and then we are building slowly. You know, because the FDA, I think, is going to get into translational software, which was actually the first decision support, they got shut down. And so the FDA is going to regulate this space. And so I think we're going to have to build.
We have a question online as well. The question is from Hila. Is PGX testing now covered by health insurances?
It is covered by CMS, and what CMS does, most health insurance companies follow. It is also dependent on states. So for example, Blue Cross Blue Shield of Alabama is more reluctant than Blue Cross Blue Shield of Minnesota. But Aetna covers the big companies, sorry. The Blues, Aetna, Optum, so Humana, they cover, several of the private companies do cover PGX testing.
Is it broadly PGX testing or it's indication specific?
So you have to, so let me give you an example. Let's say you have a patient that is getting a coronary stent and you say, I'm considering Plavix and I want to test CYP2C19. You may run the panel, but then you bill for CYP2C19. So you bill for the gene, not the panel. Let's say there is a psychiatry patient who you want to do testing because you're going to start them on Paxil. And so you do 2D6 and 2C19. Anytime you're doing more than one gene, you can bill for the panel because 2 genes is probably more expensive than the panel. So if you were going to do this, you know, we are going to roll it out after we do the ApoE and lecanemab with our Alzheimer's disease folks. We're going to roll it out for colorectal cancer. And there, they want to test in newly diagnosed patients. So you have the newly diagnosed patients when they're newly diagnosed, if they can go ahead and test, you now have the patient that's in clinic, which means you can bill under the CPT because they have to have other workup done before they are hospitalized for their chemo. And so you have time to actually do the genetics and bill for it. The inpatient stuff would just be eaten by the hospital because, you know, if the patient comes from the ED to the cath lab and the physician orders it, there is no CPT. So the volumes that are done on an outpatient basis is what will break even.
Well, I hope this answers your question. Are there any other questions online?
Can I ask one more question?
Yes.
So it's kind of related to the insurance coverage. So I guess, for those genes, right, a PGX, you only need to test once, right, in your lifetime, right? So how are you going to know, okay, these patients have that gene tested before or not? And for the insurance company, well, are they going to cover it again if these are being tested?
Yeah, so we are going to have to work through those issues. Here is our current thinking. I don't know if it is completely worked out, but let's say you have the cath lab example, patients getting stented, you do the panel, you bill for 2C19. In the future, let's say they come and they have colorectal cancer, and now you are going to use the DPYD and UGT information. I think you can bill for the panel. But then if they come to if they now have depression and you say, I want to test, I don't think you can bill because you've already billed for the panel. I don't know how a system tracks this and who is responsible for tracking it, but it is very complicated. So you can bill for single genes as you use them, is my understanding. And CMS, actually, the guidance on pharmacogenomics, if you just do CMS pharmacogenomic guidance, it's pretty well written. It's not a lot of legalese. It's clear to understand. So you and once you bill for the panel, you can't bill for anything else. Somebody needs to track that I've already built, right? So because there is only a technical fee, right? So you are being paid to do the genotyping, not the interpretation. And so if you use that data again in the future to say, here is what I would do for antidepressants, now that I've already built for the panel because you had, uracil based colorectal cancer therapies, I'm doing a consult, but I can't bill for the testing because I've already billed for it. And there is no professional component yet.
Well, I guess this is a question insurance company have to worry about, right? Because essentially you can always have double tested, labs will be happy to get more revenues. It's really up to the insurance company to track things.
Yeah, so CMS actually did a big audit of genetic testing, right? 2019, 2020, there was like a big spurt. And, that is why I think the PGX regulation is the way it's written, because they're trying to get you to think about why you're testing it and what medication, right? So you have to have that knowledge. I think that to limit or to make sure that we are using this appropriately and not just saying, well, I want, it's like me saying, Kong got an MRI, I want an MRI, and you can't have an MRI. So.
Okay, great. So it's 10 past 5 o'clock. Thank you so much for